Well, hello, everyone. My name is Michel. Thank you very much for joining us tonight, and a Happy New Year to all of you. Today, I'm delighted to host Dr. Simon Stott, who is the Director of Research at Cure Parkinson's, the well-known Parkinson's charity, and my favorite. In what is now becoming a no silver bullet tradition, Simon is going to talk to us about his main PD research takeaways of 2024 and research results he will be looking for in 2025, this new year. But before we start, as always, I would like to remind everyone that this session is for information and education purposes only. So if you're seeking medical advice, this is not the right place. Please make sure that you consult a medical professional. As always, again, there will be time for questions at the end of the presentation. And if you want to ask a question, please use the Q&A function at the bottom of your screen. And for those of you who don't know us yet, No Silver Bullet is managed by Mark Lambert and myself with the aim of sharing Parkinson's expertise. So we aim to help you and, frankly, to motivate you to make informed choices on how to adapt your lifestyle, to manage your symptoms, and hopefully to slow down disease progression. As most of you know, we post the recordings of our webinars on YouTube and Spotify, and we also post short videos on TikTok as long as we can, and Instagram. The details are available in the chat section at the bottom of your screen. So if you go to chat, you will see the links to those various media. You can also visit our website, which is nosilverbullet4pd.com, nosilverbullet4pd.com. But let's come back to today's topic and to Dr. Simon Stott. Simon, thank you very much for agreeing to speak to us tonight. The stage is yours.
Thank you, Michelle. Thank you, Mark. Happy New Year, everybody. I'm just going to share my screen. I'm assuming you can all see this.
We can see it, perfect.
Presentation mode. And I'd like to start off on this. Last year when I gave this presentation, I was giving a talk with my headphones on. I've got my headphones on tonight, and on social media, immediately after finishing, I found this.
Princess Leila.
And I have to agree, I think it's funny as well. So as Michelle was suggesting, I'm going to provide a review of some of the takeaways from 2024 in terms of Parkinson's research and what to look out for in 2025. Before I start, I just point out that I am an employee of Cure Parkinson's. Charity was founded in 2005. It's a medical research charity focused on disease modification. We do fund some of the projects that are mentioned in this talk, and where that is, for the purposes of full transparency, where that's the case, there is a small logo from Cure Parkinson's. I've tried not to be too biased because there's a lot of interesting research being done by others as well. So for tonight, with regards to 2024, we'll look at a tale of two trials, which was the lixisenatide study and the exenatide study. For us at Cure Parkinson's, this basically defined 2024. Then we'll look at some advances in symptomatic therapies with regards to produodopa, or what's now been called Vyalev, and also the developments coming through with regards to adaptive deep brain stimulation. Then we'll talk about two research reports that I thought were really, really interesting. Just my personal opinion, but maybe others will find them interesting as well. And then we'll move to 2025, what research events to look out for. Specifically here, we'll be talking about the Edmund J. Safra Act Accelerating Clinical Trials for Parkinson's Disease platform, large multi-stage, multi-arm project that will be kicking off in 2025, and also ASPRO-PD, a large phase 3 clinical trial for the respiratory medication Ambroxol. Then we'll have some clinical trials that will be coming through as well, and the start of additional studies. And we will be sure to see more data, more research being done on the biological staging of Parkinson's, and I'll explain what all that means shortly. So, as I suggested just a moment ago, the big event for us at Cure Parkinson's was the GLP-1 receptor agonist studies. GLP receptor agonists are a class of diabetes treatment, and the main one that's been tested in Parkinson's was exenatide. And in 2017, a group at UCL University published the results of a phase 2b study that in 60 people that were treated for 48 weeks with either exenatide or with placebo. And the study showed stabilization of motor symptoms in the exenatide treated individuals. You can see that across the blue line in the graph there. The blue line sort of goes down, which suggests improvement, and it stays at that level across the 48 weeks of the study, while the red line, which is the placebo group, progresses in their trajectory, indicating a continuation of symptoms getting worse. And this was a very encouraging result, and it led to the initiation of further clinical trials focused on GLP-1 agonists. And one of those was the lixisenatide study. And this was a much larger study that was conducted in France. It involved 156 people with Parkinson's, early stage Parkinson's. They were all within 3 years of the age of diagnosis. And they were randomly assigned to 12 months of lixisenatide or a placebo treatment. And what the researchers found at the end of that study was that the lixisenatide group again stabilized in their motor symptoms, while the placebo group continued to progress in terms of their motor severity. And you can see that in the graph there. The gray line is the lixisenatide group, and they're basically horizontal, while the placebo group is in black, and they continue to go upwards in their trajectory. So we've got two really interesting, really encouraging results with the GLP-1 agonists. And what's quite curious is that post hoc analysis, so this is analysis that you do of the results after the study's been published, is what they found in that post hoc analysis was that most of the effect was occurring in the younger cohort, the younger participants in the study. So those aged under 60 were having a more of a response to the GLP-1 agonist than those over 60. There was still a positive response, but it wasn't as exaggerated as those under 60. And this was interesting because in post hoc analysis of another GLP-1 agonist, a drug that was developed by a biotech company called Neurally, this drug is called NLY-01. In a 36-week study, they didn't see a positive result, except in those individuals aged under 60. So you can see in the left-hand graph, the dotted gray line is the placebo group, and the two blue lines are two different doses of the GLP-1 agonist drug, and the blue lines are well below the dotted gray line. Whereas on the right-hand graph, you can see the dotted gray line is kind of at the same level as the blue lines. So there's something really interesting going on with GLP-1 agonists, was kind of the takeaway from all of this research. And that led to the phase 3 exenatide study, which had been conducted over a 2-year period and involved 200 participants with Parkinson's across 6 sites in the UK. And on the 14th of October last year, the participants of that study were gathered together for a meeting, and they were told that the study had not met its primary endpoint. And obviously this was extremely disappointing news, not only for the participants and their families who'd taken part in this long study, but also for the investigators that had put so much effort into the setting up and the running of the study as well. The study had been conducted during the COVID pandemic, so everybody involved really deserves a medal for their fortitude and stamina in keeping the trial going. So now we're waiting on another GLP exenatide study, which has been conducted in Stockholm. This is an 18-month phase 2 study. And once we've got the results of that study, we'll be able to sit down and go through all the data and have a really good think about what next steps should take place for GLP-1 agonists in Parkinson's. We're also waiting on a lot of biochemical studies from the phase 3 study as well. And there's a brain imaging study as well that we'll gather all the results, and we'll see what it all looks like when it's pulled together. One obvious question is, what does this mean for GLP-1 receptor agonists in Parkinson's? And it is I think it's a major setback, but hopefully it's not the end of this class of drugs, because there were some really encouraging results very early on. And now I think we just need to do a bit of tweaking of the drug and see if we can replicate those results in some way or some fashion. When levodopa was first developed, it's now the gold standard for Parkinson's treatment. But when it was first developed, there was some extremely successful pilot studies followed by pretty catastrophic, much larger, better controlled studies where they just couldn't replicate the results at all. And it was tweaking of the drug, making it a combination therapy with another drug called carbidopa, that actually made it what it is, what a successful product it is today. And drug companies are already looking at GLP-1 agonists and doing a bit of this tweaking. There are some companies such as Cure Pharmaceuticals, which is developing a dual agonist. So it's targeting both. It's a drug that targets both GLP-1 receptor, but also another receptor called GIP. And they've designed their drugs for entering the brain. They've tried to elevate the levels of their of the GLP-1 agonist in the brain. And another company called MetP Pharma AG is working on a nasal delivery system for GLP-1 receptor agonists, both single agonist and dual agonists, to get them into the brain better. One of the problems with the GLP-1 agonists is not a lot of it gets into the brain. And if it needs to have that central effect inside the brain, then we need to get the drug accessing the brain better. So there's still a lot of ongoing activity, and there's going to be a lot of discussion over the next 12 months with regards to where this field goes from here. Moving on to advances in symptomatic therapies. One of the really encouraging bits of data that was presented at a couple of research conferences last year came out of the biotech company AbbVie, or the pharmaceutical company, excuse me. They have been developing produodopa, or Valiv, and it is a continuous 24-hour infusion delivery system for levodopa. So if you look at the graph on the left-hand side of the screen here, down in the corner, the purple line is standard pill form of levodopa. So when the line represents the amount of levodopa in the blood. So the purple line, you take your pill, you get a big spike in levodopa in your blood, and then it gradually goes down over the next hour or so. And by hour 5, you take another pill and it spikes up again, and then it gradually goes down over the next few hours. And by hour 10, you take another pill and it spikes up again. And this spiking is part of the problem with regards to on-off periods. And to avoid this, companies like AbbVie have developed these 24-hour infusion delivery systems of levodopa, so that you get a continuous level of levodopa in the blood. And you see it in the graph, that blue line. They start the initiation at 0 of the levodopa, and it's just pumping at a continuous rate all the way across the rest of that graph. So you get a nice standard level of levodopa, and hopefully this will avoid problems such as levodopa-induced dyskinesias. And AbbVie is not alone in the development of these sorts of systems. Another company called Neuroderm, which is owned by Mitsubishi Tanabe Pharma Group, is developing a very similar product called ND0612, and that's currently in phase 3 clinical testing. A more direct approach is being developed by a French biotech company called InBrain Pharma. They're clinically testing now. They just published phase 1 results about a month ago. And this is a direct into the brain delivery system. So there's a surgical procedure involved. And instead of delivering L-DOPA, it's delivering a very stable form of dopamine directly to the site in the brain where you need it to help with motor symptoms. And the phase 1 study is suggested as very safe and very well tolerated. And you also see early improvements in bradykinesia and dyskinesia. So the bradykinesia being inability to move, and dyskinesia being uncontrolled movements. So it's a more stable control of symptoms and motor features of Parkinson's. So that's all very encouraging. And in addition to this, you've got adaptive DBS being developed as well. There's some really interesting data now coming out, clinical data from this field. Deep brain stimulation is now quite a standard treatment for people with Parkinson's. It involves putting electrodes deep into the brain and stimulating a particular area that helps to disinhibit the motor parts of the brain. And but the problem with deep brain stimulation is that it's a continuous pattern of stimulation. It's always the same pattern. Whereas with adaptive DBS, you have a electrode recording from the brain and telling the stimulator exactly what the brain needs. So it's more of a personalized and adapted form of stimulation. It's very, very clever technology. It's all within the same package, except you have an additional electrode recording from the brain. But there's currently a clinical trial being run by the tech company called Medtronics. It involves 85 people with Parkinson's who have been implanted with adaptive deep brain stimulation, and we're expecting to see the results of that study this year. So that'll be something to look out for. It's quite exciting. Already we're seeing pilot study data though, and that's looking extremely encouraging. Despite being very small studies, this is just groups of 5 to 10 people that have had adaptive DBS implanted. The results are looking really encouraging there. It's remarkable that we've gone from painting basic pictures on cave walls only a few thousand years ago to recording and momentarily stimulating areas of the brain to help with controlling our movement. And then, of course, I was going to talk about 2 rather interesting research reports. And the first one was early in the year, a group were exploring what other functions levodopa has in the brain. Levodopa is traditionally and always been viewed as an ingredient in the production of dopamine. So you take a levodopa pill, it travels to the brain and it's turned into dopamine. It helps, and the dopamine helps to is the oil in the machine in terms of motor movement. But then with this study, the researchers decided to block the enzyme that transforms L-DOPA into dopamine. That is called dopamine decarboxylase, or AADC. And when they blocked this enzyme, they saw no conversion into dopamine. And they did this in mice. And if you look at the graph on the right-hand side at the bottom of the screen there, if you look at the red line, this is your traditional levodopa treated animal or treated Parkinson's mouse. You see a spike in the amount of activity, and gradually over time it dissipates and they go back to a Parkinsonian state. And then there's the blue line, and the blue line represents an animal that's been injected with levodopa, but also with a AADC inhibitor. So they're not producing any dopamine. And what the researchers saw was it took about 45 minutes, but the animal suddenly started moving, not just a little bit, but a lot, moving around, moving around, moving around, moving around, and it lasted a very, very long time. And the researchers were really scratching their heads, but what they found when they did a deep analysis was that there's this molecule called ophthalmate, and they're now investigating that the ophthalmate was the molecule that was causing this effect, and now they're looking at how this the mechanism behind the effect and how this could be used in a therapeutic approach. So it'll be interesting to see how that paper, how this piece of research develops over time. But it was just a very, very novel result, and a very interesting piece of research, I thought. And then the second piece of research deals with a condition called REM sleep behavior disorder. This is a condition where sufferers actually physically act out their dreams. Typically, when we sleep and we dream, our bodies go through a temporary paralysis. There are cells in the brainstem that cause us to be paralyzed as we dream. But in people with deep brain stimulation, they lose this paralysis and they will physically act out their dream, jumping about the bed, thrashing about. It affects about 1% of the general population in the US, 2% of people over the age of 50. And it's considered a major risk factor for Parkinson's, with as many as 70% of RBD patients going on to be diagnosed with Parkinson's or a similar condition within 12 years of that diagnosis of RBD. Now, there was a study that was looking at, it was a case study that was published a few years back. And in the case study, they had a gentleman with Parkinson's who had restless leg syndrome. Restless leg syndrome is where you can't control, your leg just has to move. You can't control, it's an irresistible urge to move your leg. And the researchers decided to experiment with a drug called Tangenol, which has been used in France for over 65 years for the treatment of vertigo. The mechanism of action as to how it works is unclear, but it's believed to be involved with neural modulation. So the communication between cells, just dampening things down and allowing for more clear communication. And what they found with this individual was that his restless leg symptoms dissipated, but he also had REM sleep behavior disorder. And he came into the lab and reported that his RBD had almost completely stopped while he was being treated with this drug. And the researchers thought this is interesting. So then they went ahead and treated two other individuals with REM sleep behavior disorder, and they saw their symptoms improve as well, their RBD symptoms. But this is where the study gets really got really interesting, because within 3 weeks of treatment, the RBD scores, symptom scores have gone down. But at the same time as looking at RBD, the researchers were also doing brain imaging on these individuals. And they'd been doing brain imaging for some time before they started this treatment of Tangenol. And if you look at the graph on the, well, if you look at the image on the left-hand side, panel A, you'll see across the top a bunch of black and orange images. These are DAT scans. So you're looking at the activity of dopamine in the brains of these people, of this individual, from a bird's eye view. So you're looking down on the brain, and on the left-hand most image, you see a pretty normal looking DAT scan. This is what the dopamine system looks like in the brain. And as you make your way across towards the right of these images, you'll notice that there's less of the orange in the subsequent images. So the image labeled the third of 2014 is not quite got as much orange, bright yellow orange coloration as the one from 2013. We shift to the right, and in 2019 there's less. But then they started the treatment with Tangenol, and while there's no difference really at 2022, by 2023, suddenly there's an increase in the levels of yellow and orange. And when they analyzed the images, these are the dots you see below in the graphs, you see a dip in the symbols as the imaging gets worse, but then it comes back up and increases to basically the 2013 levels in the 2023 image. And that was patient 1 on the left-hand side, and then patient 2 is on the right-hand side. And they see a very similar situation where the DAT scan before was quite poor, and then they started treatment in January of 2022, and by July of 2023, the DAT scan has improved. So this really was quite astonishing in terms of a result. And there was one major concern that was floated regarding the results. When you looked at the clinical testing of these individuals, these 2 individuals, immediately after the initiation of treatment with Tangenol, they saw a reduction in the levels of cognitive testing. And you can see this in the first graph on the top of the screen. If you look at the green line, that line represents the score on a test called the MOCA. And the MOCA is a cognitive evaluation, and it's got a maximum score of 30. And after treatment started, which is the red box, the green line enters the red box at a level of 30, and then gradually goes down to 24. And a score of 24 represents a mild cognitive impairment. And this individual was diagnosed subsequently with mild cognitive impairment. And the concern initially was that you saw a similar downward trend in patient 2. So the patient 2 goes into the treatment with the highest score of 30, and they reduced to, I think it was 27. So they didn't get diagnosed with a cognitive impairment. But subsequent follow-up studies or subsequent follow-up analysis of patient 2 has demonstrated that their cognitive scores climbed back up to 28. So it'll be interesting to see in future studies of this agent, Tangenol, whether it actually is having an effect on cognitive ability. But given the brain imaging results, you will certainly see a larger future clinical trial of this agent in people with REM sleep behavior disorder. Now the question obviously is, would this work for Parkinson's? And that is a difficult one to answer, because in REM sleep behavior disorder, people haven't really lost their dopamine neurons yet. Whereas in Parkinson's, the dopamine neuron loss is a lot more. So we're going to have to do a lot more research, firstly, to understand the mechanism of action, how this drug is having whatever effect it's having, and whether it is actually having a beneficial effect on dopamine neurons as such, or if it's doing just something with regards to the level of dopamine turnover. But you will hopefully see more research in this area, because this paper was just fascinating when it first came out. I'm going to move on now to 2025 and what to look out for. The first major events of 2025 will be big clinical trial projects starting, and the biggest one here is the project called the Edmund J. Safra Accelerated Clinical Trials for Parkinson's. This is a multi-arm, multi-stage clinical trial platform that's been set up here in the UK. Currently, when we run a clinical trial, it's a bit like building a football stadium and playing one game and then deconstructing the stadium. So you get this crazy process of building a stadium, playing a game, deconstructing a stadium, building a stadium, playing a game, deconstructing a stadium. It takes a long time between trials to actually analyze the results and get them published, and then get funding to build the new stadium. And it's just it really is quite insane, and it slows down the process of developing new drugs. So what would be better is to have a conveyor belt where you have a continuous supply of new therapeutics, and you're just continually testing them in a seamless fashion. If something works, it continues. If it doesn't work, you take it away. And that's where this design of multi-arm, multi-stage clinical trial platforms comes in. You have one placebo arm, and you've got multiple arms of different agents being tested against that arm. So you have a schematic similar to this. You'll have a placebo arm and 3 treatments. After a couple of analyses, you'll find that treatment 2 and 3 do not work. And so we remove those and replace them with treatment 4 and 5, whereas treatment 1 continues on all the way through to regulatory approval, while treatment 4 and 5 are found not to have any effect. So we introduce treatments 6 and 7 instead, and treatment 6 is found to have a beneficial effect. So we continue with that one. This is a model that has been proven with oncology. In prostate cancer, they had 3 changes in clinical practice based on the multi-arm, multi-stage platform called Stampede. And we're hoping to have the similar effect with Parkinson's. And the EJS ACT PD platform is not the only one being developed. There is a platform, a phase 3 platform being developed in Norway called Hydra. There is a phase 2 platform being constructed in France called NS Park. The Australian Parkinson's Mission have got multi-arm trials going on as we speak. And then we also have another Norwegian project called Sleipnir, which is going to be a MAMS platform for de-risking small molecule, well, for de-risking new drugs before they go onto the Hydra platform. So just quickly testing and making sure that the dose is correct and that we have the right measure of target engagement, assays, etc., in place, so that when the agent goes on to the larger clinical trial platforms, it'll be ready for prime time. And one of the more interesting projects is the P2P, or the Pathway to Prevention trial, which is being set up by the Michael J. Fox Foundation in America. This will be recruiting people who are yet to be diagnosed with Parkinson's for interventional treatment. So they're going to be trying to intervene before Parkinson's even develops in these individuals. And I'll come back to that in a moment, if you don't mind. So that the multi-arm, multi-stage platforms will be something to look out for as they develop over to 2025. The EJS ACT PD will be recruiting. The initiation of recruitment will start shortly. Another study they'll be recruiting is the ASPRO PD study. This is looking at ambroxol, which is a respiratory medication that's being repurposed for Parkinson's after it was found to elevate levels of a particular enzyme called GCase in cell screening studies, drug screening studies in cells. And ambroxol was found to be neuroprotective in models of Parkinson's, and that led to the setting up of a phase 2 clinical trial called the AIM-PD study. It involved 18 individuals with Parkinson's who were treated for 6 months. And it was really a heroic effort by the people involved, because they were taking 24 pills a day in addition to their standard Parkinson's therapy. And it's a pretty foul tasting drug as well. It leaves a very bitter aftertaste. So my hat is off to these individuals who did this for 6 months. But the results were very encouraging because we found that in all of the participants involved, the levels of this enzyme called GCase went up. GCase is an enzyme that's associated with Parkinson's, and it's reduced in people with Parkinson's. So the goal has always been to identify drugs that elevate this enzyme and test that in large studies, large clinical trials for Parkinson's as a disease-modifying therapy. And this positive result in phase 2 has led to the setting up and development of the ASPRO-PD study. It's being led by Professor Anthony Shapiro at UCL, and is a phase 3 2-year study involving 330 participants. Half of them will have a genetic risk factor called GBA1, and GBA1 is the section of DNA that produces the instructions for making the GCase protein. And the primary outcome of the study will be an improvement in motor score using the UPDRS part 1 to 3. And that study will start recruiting very shortly. In our autumn update to folks who are interested, we disclosed that there's 15 research sites that have now signed up for the study across England, Scotland, and Wales. The trial has been approved both by ethical and regulatory agencies, and subject to just some final control, quality control tests on the drug and placebo. The trial should start recruiting very early in this year. And if you'd like to register your interest in the ASPRO-PD study, you should have a look at the PD Frontline website. Part of being recruited to the trial will be a genetic test just to determine whether you have the GBA1 variant or not. It's not a inclusion or exclusion factor. It is simply to determine who is a carrier and who isn't a carrier. In addition, though, large platforms, there'll be a lot of clinical trial results coming, and there'll be some new clinical trial, other new clinical trials starting as well. One of these that is of interest is the AZA-PD study. It's azathioprine is an immunosuppressant medication. It's used in people with inflammatory conditions like rheumatoid arthritis. And Dr. Caroline Williams-Gray at Cambridge University has been conducting a 12-month study involving 60 participants with Parkinson's. And they were taking azathioprine for 12 months, followed by a 6-month follow-up period. And the results of this study should be published in 2025. And as she's finishing this study, Caroline will be turning her attention to the DAPA-PD study. This is a follow-up study that's looking at a different kind of anti-inflammatory medication. It's looking at a protein called the NLRP3 inflammasome. This is a key component of the inflammation process. Inflammation is basically the way the body tells a part of the body tells the rest of the body that something's wrong. And the NLRP3 inflammasome is kind of like a loudspeaker for that signal. Once the signal reaches a particular threshold, the NLRP3 inflammasome is activated, and it causes an immune response, an inflammatory response. And what has been found is that people with Parkinson's have got an activated inflammasome signal in their blood, or in their cerebral spinal fluid, the fluid that the brain sits in. And biotech companies have been developing drugs that dampen down or inhibit the NLRP3 inflammasome to try and counter this effect, with the hope that it's going to have a disease-modifying response. So the DAPA-PD study is going to be testing a drug called dapinsutral, which is an NLRP3 inhibitor. It's been developed by a biotech company called Olitech, and it'll be tested in 36 people with Parkinson's. They'll be treated for 12 months. There'll be 2 phases of the trial. There'll be a 6-month randomized double-blind phase in which participants will either be on dapansutral or a placebo. And then there'll be an open extension period for the next 6 months, meaning that everyone in the trial will have the opportunity to have access to the drug. And this trial will start in 2025, and the results will hopefully provide useful information for a much larger phase 3 clinical trial of this drug if it is found to be of interest. Another study that will be starting this year, and this one has kind of surprised me because it just came out today. It'll be the Expedite 2 study. This will be a phase 3, so it's the final stage of clinical testing before you go to the regulators, a phase 3 clinical trial of a stem cell derived therapy for Parkinson's. It's an allogenic approach, meaning that the cells are coming from a different source than our own bodies. But it'll be starting in the first half of 2025, and it's going to involve 102 people with moderate Parkinson's. There'll be a double-blind study assessing safety as well as efficacy and overall impact of this cell therapy approach. And it's been conducted by Blue Rock Therapeutics and their pharmaceutical owner, Bayer. And it'll be sham surgery controlled, and the participants will be assessed over a 78-week period. So that'll be a very interesting trial to keep an eye out for as it gets started. There's a lot of genuine interest from the Parkinson's community with regards to stem cell therapies, and this is representing quite rapid clinical progress or progress towards potential clinical use. Then finally, we'll be seeing a lot more research on biological staging. And by biological staging, I'm referring to the fact that when somebody gets diagnosed with cancer, a biopsy is typically taken, and the doctor is able to work out exactly what type and what stage of Parkinson's a person has using biological measures. All sorts of assays and chemical reactions can be done, and you can know quite precisely what type of cancer somebody has and how it should be targeted in terms of intervention. Whereas with most neurodegenerative conditions, like Parkinson's, a diagnosis is still based on clinical rating scales, and the staging of these conditions is very basic. In the case of Parkinson's, we have things like the Hoehn-Yahr 1 to 4 scale. But more recently, in Parkinson's research, there have been a lot of biological markers that have been identified, and these include enzymes like dopamine decarboxylase and mitochondrial DNA damage in blood. And further validation and development of these biomarkers over 2025 will hopefully help form the foundations for future diagnostic tests, as well as very powerful clinical trial tools that can be used for assessing disease-modifying therapies. And key among them has been the alpha-synuclein seeding assay. This is the most advanced of the biological assays, and also the most extensively assessed thus far. And they've been used for generating different staging systems for Parkinson's. So here's 2 examples here where 2 groups have proposed different staging systems of determining where somebody is in the spectrum of this condition we know of as Parkinson's. And this is important that we have a good biological markers and a biological staging system, because they will allow us to make more definitive diagnoses of Parkinson's or whatever the disease will be called in the future. But also it will allow us to detect it a lot earlier. and hopefully that will allow for very early interventions. So we'll be treating people before they've developed symptoms, and ideally they will never go on to develop exhibit those symptoms. So that's the big picture goal for future generations. And a good example of this is now being tried. I was talking about the Michael J. Fox Foundation's multi-arm, multi-stage platform trial called P2P, or Pathway to Prevention, a moment ago when we were talking about the MAMS platforms. They will be setting this up over the next 12 months, and they're hoping to kickstart it in 2026. They'll be starting to recruit for this trial, hopefully, but it'll hopefully catalyze the clinical development for prodromal Parkinson's, or what they're referring to as neurosynuclein disease stage 2B. It's got a nice ring to it, doesn't it? So just to summarize, I think I've spoken for too long. I've probably put half the audience to sleep, but that's okay. We'll finish up here. It was a really interesting year for Parkinson's research last year. A lot of GLP-1 receptor agonist results were provided, new symptomatic treatments coming online and further development, and some really interesting groundbreaking research studies that have provided new insights. And 2024 will be a very busy year with major clinical trial platforms being set up, and new clinical studies starting, and more research focused on biological diagnostics and staging systems. And I will stop there, say thank you very much, and I will take any questions.
Thank you very much, Simon, for this very interesting presentation. I'm particularly, well, all of it is interesting, but I'm particularly happy that you included the VLF, proteodopa, and the adaptative DBS. Those are topics we will re-explore later this year. Let's just have a look at the questions that we just received as we were listening to you. I think that actually the first question, perhaps taking them chronologically, is from Audrey, who is asking, when does phase 3 UDCA trial start, please? No, I think that UDCA was part of your research updates, not the last one, but the previous one. I don't know if you're aware of the details of the dates for the trial.
So I probably shouldn't say this, but UDCA will be one of the drugs in the EJS-ACT-PD trial. So it won't be one of the first, but it will be coming online. I think it's the third drug to come online in that study. I'm going to get in a lot of trouble for saying that.
I think we will keep it silent between us, between the 200 of us.
Yeah.
So Leah has a question about something I don't know. It's called FGF1, and she's asking whether you have an opinion on research studies being done on FGF1 intranasal delivery in the British Virgin Islands and the Bahamas.
Yeah, that's I've got to really be careful what I say here. There is very little preclinical research supporting FGF1 in Parkinson's research, and there are clinical trials being conducted, if you can call them clinical trials, that are they require the participant to actually pay a fee to be involved, and I cannot encourage people to take part in those studies. The research supporting them is very limited, and I can't endorse that sort of thing. Sorry.
Okay, that's clear enough. Thank you very much, Simon. Joseph is asking about the GLP-1 and the difference in impact according to age. And he's basically asking if you have an hypothesis as to why are the younger patients at a less advanced stage of the disease? Are the neuroprotective effects more effective in younger people? Or are there underlying types of Parkinson's that are different according to age?
So these results are fresh off the publishing press. Of the printers, we don't know. We haven't had an opportunity to look into this. It could be just down to basic metabolism, but it's probably something a lot more complex than that. I'm afraid I don't have an answer to it.
No problem. Thank you very much. Katsura is asking about GLP-1 agonists, and basically is referring to nausea problems that he believes or she believes have happened frequently. Can you talk about that? And the second part of the question is, was it a double-blind test? I think it was.
Yeah, the second part, yes, it was double blind. The nausea is quite common. Some people don't experience it, but most people with, not most people, but a large percentage of people with diabetes who take GLP-1 agonists do experience nausea. And if you look at the results of the studies in Parkinson's, a fair percentage of people with Parkinson's who are involved in those studies experienced nausea as well. There was concerns that nausea and weight loss, which is also associated with GLP-1 agonists, would be an unblinding factor in these studies. But in the first study of GLP-1 agonist in Parkinson's, the only person to drop out of the study for weight loss was actually in the control group. So it didn't have such
wasn't getting the drug.
Yeah, he wasn't getting the drug, and so it wasn't so much of an unblinding effect. But
Maybe
nausea is quite
nocebo.
common.
Thank you very much. Donna has a couple of questions. The first one is, you referred at some point in your presentation to the blocking of levodopa turning into dopamine. And she's asking if this is similar to the approach of Dr. Jonathan Zachner-Bernstein, who spoke on one of our webinars.
Yep.
She's also asking if there is update on his research.
I don't have an update on his research. I'm familiar with the work and the hypothesis, and yeah, I'm not sure that I can really answer the question. I don't have any insight there, I'm afraid.
No problem. Donna, if you're not aware of this, Jonathan Zachner-Bernstein publishes a periodic, I think it's monthly newsletter where he updates people on his progress. If you want to email me afterwards, I can give you the details. Now, second question from Donna, and she's asking if any research is being done on the drug ibogaine.
Ibogaine? No, not that I'm aware of. A lot of people ask about it, and it can have some nasty side effects, so it's got to be carefully handled. But no, I get asked about it a lot, and there's simply not enough research that has been done on it to be able to provide any sort of opinion.
Thank you very much. Another question from Joseph is asking about prasinezumab, that anti-alpha-synuclein drug. It was not part of your, well, you can't have them all, but actually, do you have a view on the potential interest of this trial?
So yeah, if you look at last year's No Silver Bullet presentation, I talked about it there, and it's quite remarkable. This is a drug that's been developed by Roche. It's an antibody. It doesn't really get into the brain, so I'm curious about the mechanism of action of the effect. But if they've done an extension study of the original trial, and everybody on that extension study has basically stayed on the drug for 4 years of extension, and the results are quite compelling. The participants all have stayed at a pretty constant level in terms of their Parkinson's. So they've done a second study called Prodova, and that at the end of last year in December, they announced that it hadn't reached its primary endpoint, but they were encouraging exploratory signs in the data, and they've started an extension arm of the Prodova study now as well. So we'll see. It'll be interesting to see what Roche decides to do next based on that program.
So Deepak was asking a question about any advancements in the use of stem cells in PD. I think you coded that one already today.
Yeah. So Blue Rock Therapeutics is going phase 3, and Aspen Neuroscience, another company, is just announced that they've finished dosing their phase 1 cohort. There's at least 4 or 5 companies involved now with different programs. It's reached a point. It used to be stem cell therapy was kind of periphery, but gradually over the last 10 to 15 years, the protocols for making dopamine neurons in cell culture have been perfected to the point where pharmaceutical companies are ready to step in and make the big investment. And you've got Bayer buying Blue Rock Therapeutics. You've got Novo Nordisk, of all companies. They did diabetes, and they've gone and purchased the rights to the STEM PD study. So they'll be taking that into phase 2 and phase 3. Sumitomo in Japan has bought the rights to the Kyoto protocols. So it's quite a lot of big pharma companies now buying into cell transplantation for Parkinson's. And it's quite exciting to see Blue Rock Therapeutics going phase 3.
Thank you very much. Now, someone is asking here about focus ultrasound and the fact that there was a trial to use focus ultrasound on both sides of the brain and not only one side, as it has been more commonly done. Do you have an update on this?
It's an amazing technique because it's instead of DBS where you go in and it's a full surgical procedure with focused ultrasound, you walk into the clinic, they zap you, and you walk out same day. But it's a irreversible approach. You can't unzap, and it's not tunable. Whereas with DBS, you can tune the levels of stimulation. So those are the two caveats with it.
Mm-hmm.
But for those who are scared of taking the DBS route, it's an option you can talk about with your clinician. And yeah, there have been some studies recently where they've been doing both sides, and the early initial results are quite encouraging.
Yes, and we had actually, I'm just trying to see when it was, we had a webinar on exactly this topic. That was last year, I think. If you have a look at our videos, you will find it actually on focus ultrasound. It was really quite interesting. And it was done in Spain. That was by Dr. Raul Martinez. So if you look up the presentation that took place 10 months ago on our YouTube channel, you will see a full presentation on this topic. Someone is asking about research done in gut-related conditions, such as the value of probiotics. Now, I saw today, I think that actually someone was commenting that Cure Parkinson's published earlier in 2024 some information about this improved probiotic trial. I don't know if you have any information on that trial or probiotics in general and their use for.
This improved study was supported by Parkinson's UK.
Oh, I do apologize. Actually, I do mix up my charities here.
That's okay. Very pleased to promote them. And we're still waiting on the results of that study. I think I haven't seen the results. Forgive me if I've missed them. But there's been a lot of small studies around probiotics, not large enough, not involving enough people to draw strong conclusions. It's more sort of hints of stuff, and it would be nice to see a really large study. And the short takeaway is that the results we've seen thus far, there seems to be improvements in issues like constipation, certainly, but we need to see much bigger studies around the probiotics.
So interestingly, 3 months ago, we had the pleasure of interviewing Dr. Philip Skeperians, who is one of the leaders on the topic of gut-brain axis and the vagal nerve. And his view, that was only his view. I don't want to be tarnishing the opinion people might have of probiotics. I'm using them myself. But his view was that food and nutrition were enough to palliate to what people saw as perception of benefits from that was just a probiotics. It was just one opinion. But indeed, I agree with Simon. We need more trials and more information, more research. The same person actually had a question about light and laser treatments like Symbix and others. I don't know if you have a view on that. I know you have touched on the topic in the past in some of the signs of Parkinson's.
Yeah, those studies are very, very small and in many cases uncontrolled or not very or poorly controlled. So it's difficult to draw any conclusions, I'm afraid.
I know that Symbix are looking to finance various trials around the world. So I haven't had an update on the latest developments, but we need to keep an eye on that company in particular. Not that their product is better, but they're investing in the research, which some of the others, most of the others don't actually.
Yep.
Valerie is asking about, she's referring to the Ambroxol trial, and she's asking if you have information about the other trials targeting GKs, especially the BIAL phase 2 trial, and also the GAIN-TX and VANQUA clinical plans.
Yep. So the BIAL trial, they've just announced that they've finished, well, the first person in their phase 2 study has finished the dosing, the treatment period, and they're hoping to have the top line results in the middle of 2026. And VANQUA has just completed phase 1. I'm not, or they're in the middle of phase 1 testing, and I'm assuming they're going to be trying to get phase 2 started before the end of this year.
Mm-hmm.
So we'll see. There's quite a few GCase activator trials or molecules coming through. A lot of activity in the GCase space.
Thomas has a question about Inhibicase and Anovis Bio. I don't know how much you know about that particular trial.
Inhibicase and Anovis are two different companies. One is Anovis is taking a molecule that stops the aggregation of alpha-synuclein, and they had phase 3 results last year. It was a small phase 3 study. It was a short study, and the results were there were some encouraging signs in the results, but there wasn't enough for regulators to do anything with.
Mm-hmm.
And then Inhibicase has got a molecule that's a c-Abl inhibitor. This is a bit like the drug, a cancer drug, nilotinib, that was tested a few years back. Nilotinib couldn't really get into the brain in concentrations that were required to block c-Abl. C-Abl is a sort of a bull in a china shop molecule in Parkinson's. It gets hyperactive and causes trouble. And these c-Abl inhibitors dampen down that hyperactivity and restore normal function. Inhibicase has got a nilotinib-like drug, but it gets into the brain, and they are currently clinically testing it, I think, in phase 2. But that trial is ongoing, as I understand it.
Thank you. When talking about Blue Rock Therapeutics and Bayer, you mentioned sham surgery for the control group. Janine is asking, how do you do sham surgery? You shave the skull and pretend you're operating, but you're not really?
It's a really boring day in the surgical unit for the doctors and the nurses, because they will go in, they will open an envelope, and it will say treatment or it will say sham. They are the only people that know what is in the envelope. And so the patient is knocked out. They will shave, they will do a cut, and they will stitch up the cut, and then they will sit watching TikTok or doing Sudoku for the rest of the afternoon, while the patient is kept under anesthetic. And there was a really remarkable story a few years back in the I think it was the Colorado cell transplantation study, where a lady went into the surgical unit basically in a wheelchair. And a few weeks after her surgery, she was up and walking around. And then a month later, she was up walking, hiking in the hills and doing all the things that she loved to do. 18 months later, she sat down with the investigators in the study. They opened up the envelope and they said, you were in the sham group. And it just shows the power of that placebo response, that placebo effect.
Yes.
Because, and I'd love to know what happened in her head at that moment when she realized that, or she was told that she didn't have the stem cells.
I
Yeah,
remember
it's a
reading
really interesting story.
some old research about the placebo effect. I can't remember who was the author. I think he must have retired, and it was maybe 5 or 10 years ago. But it was interesting to see that the placebo seemed to be generating dopamine production.
Yeah.
So
Yeah.
I could understand that someone who believes that he or she is being treated with something phenomenally successful might just generate more dopamine and stop generating it while being told that actually it was all sham.
it's why it's such a big, the placebo response is such a big problem in Parkinson's research, because that dopamine modulation is being affected.
Yeah.
So it's a fascinating field.
So Katsura has another question actually about biomarkers, and the comment being made is that if we don't compare healthy people with Parkinson's disease people, are we able to really make the most of those markers? And actually, would people who don't have Parkinson's accept to be taking part to some tests to just validate the hypothesis?
So the PPMI study that we mentioned before in the presentation, this is a large Michael J. Fox Foundation study. They've got people with Parkinson's, with specific genetic types of Parkinson's. They've got people with RBD, and they've got friends and family and others, volunteers, healthy volunteers involved as well. So you got you're looking at biological samples across a very, very broad spectrum of individuals. Most of the biorepository studies looking at blood and urine and all sorts of other samples will have controls, healthy controls involved. So that there's always the comparator.
A question here from someone who wants to remain anonymous, asking if we know if people who move on to the Prodiodopa pump or the VLF pump will still be able to participate in the upcoming Ambroxol trial, for instance, or what impact does it have on your participation to trials in general and that one in particular?
That's a really good question. So something I didn't say was that the Prodiodopa rollout has been carefully controlled by the NHS and AbbVie. They've only got 1,000 units in the UK at the moment, and it can be a tricky system to use because it's a needle that goes just under the skin, so it's a subcutaneous delivery system. But you need to be careful in how you inject yourself, and because the solution that the levodopa is in is it can be quite irritating. One of the reasons for quite a lot of dropouts in the clinical trial was because of that skin irritation around the injection site. So that's just something to consider with regards to Prodiodopa. But whether it's actually affects eligibility in some of these trials, that's a I'll have to do some homework on that one and come back to you.
Well, that's a sign of a good question. Mark and
Great
Marilyn are going back to Ambroxol and asking how was the level of dosing determined?
The level of dope that was in preclinical.
Ambroxol dosing, yes.
Yep, and it was determined in preclinical studies. There was a much higher dose than you use when you're treating respiratory conditions, and higher doses have been used in clinical trials in Japan, for example. But the dose was basically translated from preclinical studies.
Thank you. Nico is asking about the Stanford University vibrotactile glove, like the famous Peter Tass glove. Yep. Is anything known? Is it not considered a viable PD treatment? I would just say that myself, I had the pleasure of interviewing him a long time ago. And I watched one of his videos by another platform a few months ago, and there didn't appear to be anything new in what he was saying. So we would be delighted to have him back as soon as we hear that something is evolving. I don't know, Simon, if you have heard things, but I haven't.
I haven't heard anything. There's a lot of people doing it themselves.
Yes.
Making their own gloves, but I haven't heard anything, I'm afraid.
Someone is asking about, are there going to be any clinical trials of ibogaine for Parkinson's in the future?
We've looked at the molecule for our International Link Clinical Trials program, and we haven't taken it forward. There's just simply not enough data to justify taking it forward.
Thank you. So Ken is asking about, is there any research coming about PD that may start from intestine or the digestive system? So Ken, I would like to refer you to the extensive webinar we did 3 months ago with Dr. Philip Skeperians, who spoke exactly about that. And he spoke about the importance of the gut-brain axis. So you can find a lot of information there as a great starting point. Andy is asking if you have a view about the use of sham surgery for the US test. I mean, what is your reaction? You spoke about the reaction of the surgeons that day who were opening the envelope, but in terms of like the patient perspective.
Yeah, it's a massive ask of the participants. There's been a lot of discussion around it. You can have someone like Professor Roger Barker come and talk on your
He's scheduled for the end of the year, by the way. He's
yeah,
scheduled
so
for the end of the year.
you can ask him the same question because he's been central to some of those discussions, and he's probably the better person to ask that one to.
Thank you. Harry is asking about. Yeah, well,
I'm throwing Roger under the bus there.
No, I'm sure that actually that's a bus he likes to be under. It's probably not a problem for him. And we will have, I can't remember exactly what month we have him. I think it's in November or December that we have lined him up. He wanted to be lined up towards the end of the year so that he can have more research by then to present to our audience. So we wanted to have him back. Harry is asking about the treatment you mentioned for REM disorder and asking if it's likely to be available in the clinic soon. I think, Harry, the first step would be to do a trial, a medical trial of this, because only 2 people were basically had data presented so far. And Simon highlighted some of the potential downsides. In particular, I think, what was it again? Yeah, people cognitive capacity. Sorry,
Well, yeah,
I'm losing my own cognitive capability just talking about it.
Yeah, there's only a couple of data points there, so they were just trends that needs to be better investigated. And Tangenol's only indicated in France. You can't. There's no other jurisdiction for it, so it's not allowed to be used off-label in the UK, for example, or the US. There is a biotech company, InfraBio, who's developing a molecule very similar to it, and that could be something to watch. But yeah, at present, as you say, it's only in RBD, and a couple of individuals with RBD, and a lot more research is required.
So an interesting question here, actually, slightly, well, it is on topic, but slightly different angle than all the other questions. Someone is asking about research misconduct by Eliza Maslia papers on the topic of alpha-synuclein. I'm not aware of this, but I will definitely have a look afterwards. How may it affect future studies on alpha-synuclein research?
Yeah, so this is one of the godfathers of the field of synuclein research, and it caused. It was at a large conference when the news came out, and it caused everybody was so shocked. There's a very human side to research where people are going to exaggerate a particular result, and maybe it's just for the personal ego's sake, or maybe it's for career progression. There's all sorts of reasons why, but it's a problem that pops up every now and then, and it was really shocking that in this particular case that this individual had been involved with a pattern of it.
Does it invalidate what we think about alpha-synuclein so far, or not?
No, synuclein has been looked at by every man and his dog since, and there's so much research now being conducted on it that it doesn't really invalidate it at all. There's so many people that have replicated the result, or yeah, it's
not impactful in terms of research on Parkinson's.
No, it's like one member of Manchester United cheating. The other 10 players are still going to get the same result, right?
Perfect. Okay,
I'm not a Manchester United fan, by the way. Ipswich Town.
You can apply this to your own location and favorite sport. So Greg has a question about something I don't know. It's called Wegovy. And I don't know if you're familiar with this. He's basically asking if Wegovy is being prescribed as an approved Parkinson's treatment. I've never come across that.
Wegovy is a GLP-1 agonist, And one of the things I should have said with regards to the GLP-1 agonist research is that these drugs are designed for people with diabetes, and they've now been indicated for weight loss. And if you take Wegovy, it doesn't really get into the brain very well, and it does cause weight loss. And people with Parkinson's can't really afford the weight loss part. So we have to be cautious with regards to GLP-1 agonists in terms of that weight loss issue. But Wegovy has not been explored thoroughly enough in Parkinson's for us to have an opinion on it, or to make a judgment on it.
Simon has an interesting question about lixisenatide and exenatide. And he basically is asking whether the data will be pooled at some stage, or has it already been pooled to some extent, to try to see whether by putting two pools together, you might have a bigger pool with better quality information, perhaps.
Yeah, it's a bit difficult to compare the two because they're slightly different drugs, GLP-1 agonist drugs, but there are post hoc analyses going on at the moment that are comparing the various clinical trials that have been conducted, looking for where there are similarities and where there aren't. And over the next few months, there'll be meetings to review the exenatide study. Once the Stockholm study results are available, the experts will get together and start pooling the results and comparing them and coming up with new hypotheses and what the next steps are. It's just slightly too early at the moment.
Understood.
And just to add, with regards to the Wegovy question before, I forgot, there is a Japanese trial at the moment that's looking at Wegovy in people with Parkinson's, but that's only just only just underway. It's maybe 6 to 12 months in, so it's still got some time to go.
So thank you, Simon. Given that we may not have enough time to answer all questions, I will sometimes refer some people to previous presentations instead of asking the question to Simon. So there's a question about mitochondria and potential effect on PD and progression. I'd like to refer that person whose initial are SC to a presentation by Sonia Gandhi that took place 5 months ago, and the video recording is available on our YouTube channel. Someone is asking here about your view on information on the Bhutanetap trial by Anivis Bio that took place in late 2024. Is it promising, according to you?
So this is the Innova study. This is the phase 3 study that I mentioned before. That was too short, and some interesting exploratory analyses, but they'll need to do more research, unfortunately.
Thank you, Simon. Lisa watched with interest our Dr. Dorsey interview a month ago, where he was talking about his view on the causes of Parkinson's and how to prevent Parkinson's. And she's asking, quite interestingly, what, if any, research that you know of is being done to get us closer to understanding what causes Parkinson's? Because we talked today about symptomatic and disease-modifying therapies, but we didn't really talk about fundamental research to understand how to stop it in its infancy.
Yep. So there's a lot of research going on in this space. The most interesting research is looking at not Parkinson's as a whole, but looking at the different subtypes of Parkinson's. It is possible that there are people with Parkinson's that have a very vulnerability with regards to mitochondria, and they might be more vulnerable in terms of exposure to things like pesticide or air pollution. And then there'll be people who have got a vulnerability in the waste disposal system of cells, and they might be more vulnerable to other things like a viral infection or something like this. There's a lot of research going on looking at the causes of this condition we know of as Parkinson's, but I would say that the most interesting of that research is focused on the different types of Parkinson's and breaking that down and hopefully identifying the causal factors.
Thank you, Simon. Stefan is referring to the Blue Rock Therapeutics Bayer stem cells study. Of course, he's aware, listening to you, that there's a phase 3 study coming up, but he's asking whether you could elaborate a bit more about the results of the phase 2 study that came before that and how positive they were.
Yep. So they've just recently presented the 2 year data, and it looks very encouraging. The worry was with this approach of growing dopamine neurons in cell culture is that every now and then you'll get a cell that's transplanted that's still a stem cell. And if it's a stem cell, it's just going to divide and divide and divide and cause a tumor. We haven't seen any tumors so far, or any signs of any cancerous sort of activity. So the teams involved have been fantastic and very vigilant in that regard, and they've got a very good protocol. And the brain imaging is suggesting that the transplants are producing dopamine, so that's a positive sign. And they're also seeing some positives around symptoms as well. And I can't remember if they presented dose of dopamine treatment data, but the most important takeaway is that it's safe and well tolerated. We're not seeing dyskinesias as a result of the transplants. The problem with it thus far is that you don't really, with the cell transplantation approach, you don't see the effect for about at least 18 months to 24 months because you transplant in a baby, a progenitor dopamine neuron, and it's got to grow, and it does this over not a few days or a few weeks, but years, and reach out and contact the cells around it, and then produce dopamine, and that takes time. So in about it takes in the 3 to 5 year time frame is when you start to see whether the transplant is actually having any actual effect.
Thank you, Simon. So a comment to our audience. I have mentioned several YouTube recorded videos of our previous webinars. Mark has very kindly added the direct links to the chat. So if you want to visit the chat section at the bottom of your screen, you'll be able to see the links to those presentations. There's an interesting question here from someone anonymous who is asking who is referring to the levodopa 24 hours infusion. And he's asking, or she's asking, whether time release levodopa can be a possible in-between solution, controlled release or time release, as we call it differently.
I'm not sure I fully understand the question.
I think I understand. I think the answer is probably no, but I think that actually I'm using both immediate release levodopa and controlled release or time release. The time release I'm using at night, it's basically the effect is less intense, but it is more prolonged. And I think the question here, could you try to cobble together several time release pills to try to have a smoother effect similar to the line achieved in the Produodopa product compared to the spikes that we saw in the immediate release?
Yeah, I'm just a research scientist. I'm not a clinician. You're probably best to speak to a clinician with regards to that sort of a question.
I would say from a personal experience, sorry, I don't want to be making it a me kind of session, but I think that the effect of the control release is indeed lasts a bit longer, but it is basically lower and less intense. So as far as I'm concerned, I suspect that actually the control release does not provide people like me at least enough boost to be an appropriate replacement to the Produodopa pump. That's my own opinion. Someone here, Michael, is referring to something I'm not aware of at all, which is ferroptosis. And he's asking whether there is a link between PD and ferroptosis, and is there any research on this topic?
Yes, so ferroptosis is a kind of cell death, programmed cell death, and there are a number of research groups looking at inhibiting ferroptosis as a therapeutic approach for Parkinson's. At present, the data the data linking ferroptosis to Parkinson's is still a bit it still needs to be strengthened. We need more research at looking at postmortem tissue, for example, identifying therapeutic cells. But there there has been research on this, and there are biotech companies developing molecules to block it. So once there's a green light in terms of ferroptosis being an important part of the pathology of Parkinson's, then those drugs will hopefully be available for testing.
Thank you, Simon. Joseph is asking about GDNF and new delivery methods for delivering GDNF to the brain with gene therapy. He's mentioning AB 1005, which you may be aware of, which is recruiting for phase 2, he tells us.
Yeah. Yep. So this is another, the pharmaceutical company Bayer. They bought Blue Rock Therapeutics, and they also bought another company called Ask Bio. And Ask Bio have been developing, they've been developing a gene therapy approach for GDNF. And then there's a phase 2. They've done phase 1, and the phase 1 results look encouraging. So they've got a phase 2 study going on at the moment. Bristol is in the UK is one site for for that study. There's also a site in Poland. I can't remember off the top of my head where else they're recruiting. Pretty sure in St. Louis, Missouri, there's a site there as well. But it's a global study, multi-center, multinational study. And Ask Bio is the company that's running it, and that, as I said, is part of Bayer, the large pharmaceutical company.
Thank you, Simon. Jeffrey has a question about something called the product called Therazosine, which is an older prostate treatment drug, and whether you're aware of any potential beneficial impact it might have in slowing down the progression of Parkinson's.
Yep. So there's a group in the Midwestern America that have been working on this, University of Iowa, and they've been repurposing this molecule. They've done a couple of phase 1 studies, and they're now looking to get it into phase 2 testing. And there's been epidemiological data. This is where you take a large population of people and you look at who's been taking the drug and who hasn't, and what impact it has. And that's been quite encouraging in terms of people taking Therazosine not developing Parkinson's. And I'm racking my brain here.
Yeah.
And I'm sorry, I'm a bit under the weather with sniffle, so I'm struggling
Not
here.
worry. I think you've already answered quite extensively.
Yeah, it is being looked at as the question. Michael J. Fox Foundation funded a phase 1 study in healthy volunteers, and that gave us a lot of biomarker data. And they've been testing it in people with Parkinson's. There was a concern that it would have a blood pressure issue and people would be fainting, but it wasn't the case. So that's encouraging. And now there are efforts to take it into next stage of clinical development.
Thank you. I have a couple of last questions here. They're a bit, I would say, technical because they refer to specific therapies or treatments that you may be familiar with. Someone is asking about updates on Gain Therapeutics GT02287. I don't know if that rings a bell. I know that Gain Therapeutics is a name I have seen several times.
Yeah, Gain Therapeutics has got a GCase activator, and that's in clinical testing at the moment. It's in phase 2 at the moment, I believe.
Thank you, Simon. Look, I think that actually we will stop here. It is 9 o'clock, and we have answered close to 40 questions. We have left 9 questions open. I think it's pretty good stats. And Simon, I would like to thank you for being with us at the start of the year for 3 times in a row now, I think. As I said, it's becoming a tradition, and we're looking forward to 2026, which will also be the year of going to Phoenix, I think, for the WPC. But thank you very much, not only for what you do tonight for our audience, but also for the work you do for the science of Parkinson's, your newsletter, which I always read with a lot of interest, and I recommend to everyone. So if you're not familiar, you want to say a word about that? Because actually there might be people listening to us who are not familiar with that, and you might just want to point them to your platform.
I haven't had much time for it over the last 12 months. I'm trying to catch up at the moment, but it's just where I take a piece of research that I think is interesting and try to explain it in plain English that my mother can understand. Yeah.
I think the fascinating thing about the signs of Parkinson's is that indeed you're using plain English, but you're not basically losing any of the critical information. So it is a serious level research information put in words that everyone can understand. So I would highly recommend it. So thank you very much, Simon. Thank you again. And to our audience, I wanted to say that thank you. Our next session will be taking place on the 10th of February, and we will have the pleasure of having Dr. Rafael González Maldonado, the well-known Spanish neurologist who is a specialist of mucuna, who will be talking about mucuna and the use of mucuna in Parkinson's. We had a presentation a few months ago about using mucuna for low-income countries. The next session will be based on using mucuna for all of us and seeing how to do it, whether it makes sense, how can it be done safely, what are the benefits and the potential downsides. So join us on the 10th of February. The invitations will follow in 10 days from now. Simon, thanks again. Thank you very much. Thank you to our audience and Happy New Year to everyone. Thank you.
Thank you. Thank you, everybody.